This section deals with the types of lab tests that you would do to distinguish between enteric bacteria versus non-enterics and between different genera and species of the enteric bacteria themselves, corresponding to exercises 46 through 49 manual. Exercise 46 deals with isolation of the enterics, and first off, what is an enteric bacteria? It's gram-negative. They're bacillus. They're non-spore forming. They are commonly found in the intestinal tracts of animals, including those of humans. And so we can use different selective and differential media to try to isolate them and try to identify them. Two of the most popular mediums that we'll use is MacConkey's agar, and then also the eosin methylene blue agar, which is referred to as EMB. The MacConkey agar is selective and differential. It is selective in that only certain organisms will grow on it, and other organisms will be inhibited from growing on this agar. Typically, it's grown or it's made as in a petri dish. It is selective because it contains biosalts that will inhibit the non-enteric bacteria, and it also contains crystal violet dye, which is inhibitory to gram-positives. So no gram-positives will grow, plus the non-enterics will not grow. Only the enteric bacteria will grow on the MacConkey, so why is it differential? Well, you can distinguish between the lactose fermenters and the non-lactose fermenters. They will both grow, but if it is a lactose fermenter, the colonies will be red pigmented. If it is a non-lactose fermenter, the colonies will be colorless. To see, you can look at the video links where the results are shown. The eosin methylene blue, or the EMB agar, it is selective in that it has two dyes in it, eosin and then also methylene blue, and both of those dyes are inhibitory to gram-positive bacteria. How is it differential? It depends on the colors of the colonies. Once again, you look at that to distinguish between them. Non-lactose or non-sucrose fermenters are going to be colorless. If it is a lactose or sucrose fermenter, then the colonies typically will be a very dark color, like a dark purple or even black colored colonies. Now there's one species that has a very unique, distinctive coloration to it, and that is Escherichia coli, commonly referred to as E. coli. It has a very bright metallic green sheen to it. So if you have an EMB plate and you see this shiny metallic green color, right away you know that that is E. coli that is growing on there. It's EMB is often used as an indicator to test for E. coli. E. coli, because it's commonly found in the intestinal tract, is used as an indicator organism for water quality. So if you're testing a water supply, wondering if it's okay for human consumption, if E. coli is in there, you grow it on an EMB plate, that E. coli is going to be shiny green, which is going to translate to don't drink the water. Exercise 47 is the TSI agar. This is the triple sugar iron. The three sugars that are present in this is glucose, lactose, and sucrose. The other thing that you can test besides the fermentation of those sugars is for the production of hydrogen sulfide and also for gas production, specifically dioxide. This is a slant, and after you inoculate and let it incubate, you look at the color. There is phenol red in it, so you can see a color change by change in pH. If both the butt and the slant are yellow, that is indicative of glucose fermentation as well as fermentation of one of the other two sugars. So you had glucose and lactose or sucrose fermentation occurring. If the butt is yellow but the slant is red, then that means there was only glucose fermentation. If both the butt and the slant remain red, there was no fermentation whatsoever that occurred. If the butt is black, then that is a positive test for hydrogen sulfide production. It combines with the iron that's in the medium and it forms this black precipitate. So you can see a black color, but the black typically is only in the butt region of the tube. If there are cracks or fissures, that's indicative that gas production did occur, so it'd be positive for gas production. During the fermentation process, the gas produced typically is carbon dioxide. Sometimes enough gas is produced that the media that was resting against the bottom of the tube is actually raised or pushed up some. Once again, we have under video links the results of them and see them. Exercise 48 is the IMVIC test. Stands for indole, methyl red, Volks-Prosker, and citrate. Also shown under video links. The indole test, what you would do is you would add 5 drops of Kovacs reagent or the indole reagent excuse me, to a SIM tube or a trypticase soy tube. If on the upper layer there, if you don't mix it, you just add the drops on top. If that upper layer turns red, it's positive. If it remains colorless or pale yellow, then it is a negative result. The methyl red, you would add about 5 drops of methyl red to MRVP tube. MRV2 is methyl red Volks-Prosker tube. If it turns red on the upper layer, it is positive. If it remains colorless or pale yellow, that is a negative. The Volks-Prosker or VP tube, for this one you would add 10 drops of the VP A reagent and then 10 drops of the VP B reagent to the VP tube. And just as a quick reminder, it has to be a separate tube. You cannot do the methyl red and VP on the exact same tube. It's the same media, but you have to have one for MR, one for VP. If it turns red, it's positive. If it remains colorless or pale yellow, once again, it is negative. And finally, the citrate portion of this test, you would have inoculated a 7 citrate tube. Normally, it is green. If it remains green, it's negative. If it turns blue, it is a positive test. All of those previous tests are under the video links if you wish to see the tubes inoculated and see what a positive versus a negative actually looks like. Exercise 49 just shows that we've been telling you often how to run these tests individually so you understand what is going on. In reality, in a lab, we don't have space for all these various petri dishes and test tubes to be incubating for the number of samples that would be coming through. So a couple of different companies have developed a rapid identification process for the enterics. These are geared towards enterics only. They do have them for some other organisms as well. Probably the most common one is called the entero tube too, and as you can see in this picture, in the back is an actual tube, and then the diagram showing a negative result and the positive result. So what you would do is there's a metal rod that goes throughout the length of this tube, and as you can see, the tube has different chambers for the various tests. So instead of individual test tubes, it's all on this one tube. You would grow your bacteria on a plate, and on an individual pure colony, you would unscrew one end of the tube, and there's a metal rod. You would touch that in onto the pure colony. Unscrew the other end of this tube, and you pull the metal rod all the way through the tube, thereby inoculating each of those chambers. Dispose of the metal rod properly. Screw the caps on the ends, and then just incubate this tube. And then what you do is, after you incubate it, you then record the positive and negative results for each chamber. And there's a way that it gets numbered, and then you come up with a number and check that against your reference book. And then it will tell you like a 98% chance this is E. coli. And this is just another one of these types of rapid test. It's an API strip. It's the same type of thing, only this one, the media is dehydrated in there, so you have to grow culture. It's easiest to grow it up in liquid broth, and then just put a drop in each one of these chambers. Once again, you would, after incubation, look and record the positive negatives, gives a number, and then look at your reference book for that as well.